Hello. In the last lecture, that is in the last video, we have covered up biomolecules. That is one of the biomolecules, lipids. I think you understood regarding the lipids that they are the 5% of the dye mass of the cell. And the next most important sort of the biomolecule that we are going to cover up today in this video is going to be carbohydrates. Okay. These carbohydrates are mainly a central idea for the metabolism in the body. Okay. And they are the energy sources. Okay. And I think you people have learned about or listened about the adrenaline gush during dangerous situations, or when your body is kept in a mode of what you call is a danger that you're forecasting. What happens is levels of the glucose increases in your blood, and this is just because the glycogen, which is a form of a stored energy molecule, is going to be converted into what you call is glucose, and the glucose levels in the blood are going to spike up. Okay, so we need to remember that if a patient is like if a patient is energyless, so the thing that we usually see that our doctors are putting them on the glucose, right, is just because glucose provides an instant energy to the patient. Okay, glucose instantly is going to participate in the cellular respiration process, whereby we convert our glucoses into the ATP molecules, which is the chemical energy that is being utilized by the body. Okay, I think you're understanding. So the biomolecule today that we are going to learn is going to be carbohydrate. So I'm specifying what are the important functions of the carbohydrates. And I even told you that it is going to cover up 25% of your cell mass, and it's going to be in your extracellular matrix. It's going to be inside the cytoplasm, and your glucoses, that is your carbohydrates, are going to be clubbing up with the proteins to form glycoproteins, and that are going to be on the surface of a cell, which are usually used for the cell signaling. Okay, and we have this carbohydrates inside the cytoplasm, which are going to sometimes be clubbed up with the lipids, as well as sometimes clubbed up with the proteins to form glycoproteins. If they're clubbed up with the lipids, we call them as glycolipids. They are being sent from cytoplasm to that of the nuclei in order to send the signaling. Okay, so we are trying to specify or know the functions of the carbohydrates. I'm telling you that these are going to be the important constituents. Like in the last video we spoke about the cholesterol being one of the component of your plasma membrane, and we saw that the buildup of the plasma membrane is just because of the bilipid layer which has been formed because of the phospholipids, where one of the fatty acid chain was being substituted by the phosphate molecule, which is a polar molecule, making the phospholipids phospholipid head as what you call is hydrophilic in nature. In the same way, now we are specifying about the glucoses, and sorry, we are specifying about the carbohydrates. Carbohydrates are also one of the main constituents of your plasma membrane, where your phospholipid, that is lipid, is going to be clubbed up with your carbohydrate over the plasma membrane to form glycolipids. And even in the extracellular matrix, we can see the presence of the carbohydrates in form of glycoproteins, and the specific biology which deals with the chemistry of these glycoproteins is going to be glycobiology. Okay, and even in the cytoplasm we see that your carbohydrates are clubbing up with proteins. They club up with the lipids to form the glycolipids. Okay, so these all, like the glycoproteins and the glycolipids, as well as the glycoproteins over your what you call is cell plasma membrane, forming glycolipids. These all are going to be the macromolecules which are going to help you in signaling. As we came to know that your cells which are adjacent in a tissue, they try to communicate with each other, and the communication is going to be successful, is going to be actually accomplished by the presence of these molecules, like these glycoproteins, glycolipids. Okay, these are the ones which are going to be. And besides putting the biochemistry of the cell, regarding, say, speaking about these what do you call it, signaling, we have a most important thing, that these carbohydrates are going to actually help in your central metabolism. As I told you that it is going to be the speaking about the process of cellular respiration, where instantly the glucoses are being oxidized into glucose are being oxidized into these ATP molecules which provide you energy. And as I told you that when an organism is in a dangerous situations, the stored form of energy which is being called as glycogen. Before I explain you this, I think I have to speak about that. Glucoses are being stored in your body in form of glycogen, which, so glycogen is regarded as a storage energy molecule, which is stored inside your what you call is liver, as well as it is stored inside your muscles. And they are being stored for the future use. And when a person is fasting, or when a person is going to be in the dangerous situations, the glycogen stored inside your liver, or stored inside the muscles, especially in the liver, is going to be converted into glucoses. And the glucose is going to be sent into the bloodstream, such that you will be instantly being provided with energy, that is ATP molecules, by using these glucoses in the cellular respiration process. Okay, so your glucose is also a sort of a monosaccharide, or it's going to be a carbohydrate. So that when we are talking about the energy, we have to think about the glucose molecules. So energy sources are the carbohydrates, and the most important constituents of your plasma membrane, extracellular matrix, as well as inside the cytoplasm for the signaling sakes, is going to be the carbohydrates. And these carbohydrates are the ones which are going to be actually stored for the future uses in form of glycogen. And even in plants, we see that the glucoses synthesized during the photosynthesis process are being stored in form of starch and celluloses, where the starch is being stored in the roots, or it might be in the sort of seeds of the plants which will be used by them, or the ones who consume them are utilizing this starch, and later it is going to be converted into the simple monomers, that is going to be glucoses, and used by that specified organism who's consuming them. Okay, so remember that we are using the carbohydrates, that is especially the category of glucoses, very instantly, making them get participated inside the cellular respiration and releasing ATP molecules for you. Or we are going to store these glucose molecules in form of glycogen, and store it in the liver or in the muscle cells. And when required by the organism, it is going to be converted into glucose, and it's going to be given off. And usually call these situation or condition where the glycogen in a dangerous situation getting converted into glucose and utilized by the organism is going to be an adrenaline gush. Okay, because adrenaline gush is going to be seen only during when the person or an organism is going to be kept in a very typical condition where it's not normal, and where it's been in a very what you call is regarding it as a dangerous situation where it's not as normal to it. Okay, so not only as the storage molecules, the carbohydrates have been stored in the body for the future use, but instantly being used, it's used in the biochemistry of the cell. Besides that, carbohydrates are the ones which are going to undergo hydrolysis process, as well as these are the ones which are going to actually release the carbon atoms, where these carbon atoms will be utilized for the buildup of another or the other sort of molecules. Okay, remember that. So they are acting as a sources for the, what you call is, sources of carbon atoms for the formation of the other molecules as well. So carbohydrates are used as a sources of energy, are used in the cell signaling processes. They are used as a sources of the carbon atoms for the formation of the other molecules. These are the ones that you can remember. Remember that your genetic material, that is going to be your DNA, as well as the other nucleic acid, which we are going to specify as RNA, which has been called as a ribose nucleic acid. These both of the nucleic acids are the ones which are comprising of the most important hexo sugars, where hexo sugar stands for the sugar comprising of 5 carbon atoms. So the ribose sugar and the deoxyribose sugar in the DNA, as well as RNA. That is, so we see the ribose sugar in the RNA and the deoxyribose sugar. That is 2-deoxyribose sugar in your DNA. These two of them are the hexo sugar. So we need to remember that. So carbohydrates are even becoming the important constituents of your nucleic acids as well. So I think now you have understood that how important is that. Not only this, but also there is a utmost important function of these, because they are going to act as these surface proteins, where I spoke about the plasma membrane, where we have these glycolipids. We know that the phospholipid bilayer is going to be having the lipid, right? So that lipid is going to be connected with the carbohydrate, where we call it as a glycolipid, which are going to act as antigens. So by listening to the terms antigens, almost I think the students are getting the recap of the blood groups that they have learned. You remember that RBCs are going to be having antigens over their surfaces, which are responsible for the ABO blood groups. I hope you are getting it. Okay, so these antigens, which are the glycolipids, I think you're getting it back again. Important. So I spoke about the plasma membrane having the bilipid layer, the phospholipid, where the lipids are going to be clubbed up with the carbohydrate to form the glycolipid, which can also be regarded as a antigen. And these antigens are ones which are responsible for our ABO blood groups. Okay, I think you have understood that. Your carbohydrates are 25% of the dry mass. They have wide ranges of functionalities. Remember that when we were speaking about the fats, we could see that they were having, like when I spoke about the saturated fatty acids, I have shown you an image where you had this. They are completely saturated with lots and lots of hydrogens. Okay, and they had only the single bonds between the carbon and the hydrogens. But remember, coming on to the carbohydrates, these are the ones which are going to show you many hydroxyl groups attached to the carbon. So once when you're going to look at a molecule having only carbons and hydrogens, they are going to be categorized as lipids. And when it is going to be the carbohydrates, you have to remember that the carbons are abundantly present with the hydroxyls, and actually this hydroxyl is going to be the one which is going to be polar, making the carbohydrates hydrophilic in nature. You need to remember this. Okay, so when there are carbons and hydrogens, they are going to be the lipids. And if they are going to be carbon rich with the hydroxyl groups, so, and if these hydroxyl groups are going to be the polars and they are the hydrophilics, making your carbohydrates to get dissolved in the water or get solvated. And these carbohydrates usually are going to get existed in two forms. One is going to be a linear form and the other is going to be a ring form. Okay, they are in an equilibrium state. They can be either in the linear, that is a linear straight chain, or they will be turning into a ring structure. And usually carbohydrates, when you are going to dissolve them in the water, that is in the aqueous solutions, we see that these carbohydrates are going to be in a ring structure. Okay, I think you have got a preview of all the uses and why there is a need to learn about the carbohydrates, and especially speaking about the last one that I was talking about was about the surface antigens, that is a glycolipids present, and which are specifically giving the A, B, O blood groups for these RBCs to the different individuals, right? We have these A blood group, B, AB, and O. Put aside the rhesus factor, but now I'm only speaking about these, what they call is RBCs having the A antigen, the B antigens, the AB antigens, and sometimes the blood group RBCs which do not contain any antigens coming under the category of your O blood group, which makes it a universal donor. Today we are going to talk about one of the specific points about these blood groups and what is a new that is the research that's going on in the University of Columbia to convert the blood group O, conversion of the other blood groups like A, B, and AB into what you call is a blood group O, such that all the other blood groups, that is A, B, AB, can be converted into blood group O by removing one specific carbohydrate which is present as a glycolipid on this A, B, and AB, but not present in A O blood group. So we can just remove it off, slice it off, and go with the formation of blood group O. That I'm going to talk regarding this at the end of the video. But just now, I'm going to start now about the carbohydrate structures and what are the classification of the carbohydrates, because specifically you are going to learn these in your syllabi, and these are important for your examination point of view. I would love to share my screen with you. Okay, and let me start with this carbohydrates. Okay, so coming on to the carbohydrates over here, the first thing that I would like to show you is going to be the classification of these sugars. Okay, the carbohydrates, and remember that your carbohydrates are actually being called as hydrates of carbon, hydrates of carbon, and the simple and the most abundant simple sugar is going to be glucose. As I told you that these are the ones which are going to actually give you instant energy by participating in the cellular respiration process, and they release ATP molecules for you. Okay, so those are the glucoses. Remember that almost all, if you are going to check this particular classification of the carbohydrates I'm talking about, they are they are classified into some categories like monosaccharides. Okay, monosaccharides. The other is the disaccharides and the polysaccharides. Before I jump into the disaccharides and polysaccharides, do you remember in the last video when I spoke about the polymers? Because I told you that biomolecules are often polymers. They are polymers. So you can see monosaccharides and not the polymers. So that's the reason I have used them often, because here now you can see that they are polymers having a very huge like repetition of these monomers for many times. Okay, so we spoke that for the formation of these what you call is polymers, there are two sets of reactions that we could see. They were none other than the condensation process, and the other was what you call is a hydrolysis process that we could see. And first, coming on to the condensation process, we'll be seeing here, I'm giving you one example of formation of this what you call is a disaccharide here, where you can see this is a monomer, that is a glucose. Okay, glucose, and this is also monomer, that is glucose. They are getting together, and these hydroxyl groups you can see over here, one of the hydrogens and the hydroxyl groups from this monomer, they are going to be removed off. Okay, so that is going to be removed in form of water molecule, and these two monomers are going to be clubbed together with the covalent bond. You remember I was talking about the importance of the covalent bond even in the lipids too. So this is also covalent bond, and we call these bonds in the sugars as glycosidic bond. So by the combination of the two monomers, that is a glucoses, we could get this disaccharide, which is being called as a maltose. And this disaccharide, because this carbohydrate is comprising of 2 units of monosaccharides, 2 units of monosaccharides, so we call them as a disaccharide. So this is a condensation process. This is a condensation reaction which is involving in removal of a water from the respective monomers and joining them togetherly with a covalent bond and forming a disaccharide like maltose. Okay, this is one of the examples. So I think I made it clear for you children that the carbohydrates are also often polymers. They will be undergoing condensation process by removal of the water molecule and they form disaccharides. So now I'm coming back to this particular classification here, where you can see clearly that the first category of simple sugars is monosaccharides, where saccharin is going to be a sweet. Saccharin is going to be sweet. Okay, so this is a sugar. Saccharin is a sweet. So mono, because it is having a single, okay, single what you call is the unit. There is they are not the polymers. Okay, now coming on to glucose, fructose, and galactose. The most common sugar that is going to be the monosaccharide, sorry, is going to be glucose, and the other one which is found in the fruits is going to be fructose, and the other is going to be galactose, which is one of the compositions of the lactose sugar, which is a milk sugar. Okay, I will give you a shortcut, the way to remember this names of the monosaccharides. You remember the gal, so usually call it a GIRL, girl. So it's going to be GL, girl, girl. Oh, girl. I think you have that such sort of a song in Bollywood. So a girl is the girl, girl. Girls are going to be the girlfriends. So it's going to be GF. So girl is going to be galactose, and the GF is going to be the glucose as well as the fructose, which is going to be. I hope this gives a sense for you to just remember. Please take it as comically. Okay, so monosaccharide examples are going to be the gal, GF. It's going to be glucose, that is fructose, as well as fructose, sorry, as well as galactoses. Okay, this is a monosaccharide. Remember, all the monosaccharides are going to have a same chemical formula. They are going to have a same chemical formula. Let me show you that. Let me show you. So you can see that these are having a same chemical formula of C6H12O6 that we are going to see. Okay, C6H12O6, but I think you can see that though they have a same chemical formula, the way of arrangement of the atoms in this particular molecule is getting differed. You can see the arrangement of these are getting differed. So because of the difference in the arrangement of the atoms here, the monosaccharides are going to have what you call is a different structure, different property, because in the last video I specified that as the structure changes, the chemical properties are going to change. So you can see that monosaccharides, though they are going to have a same chemical formula of C6H12O6, since they are going to differ in the arrangement of their atoms, they are going to get different structure. That is, they are going to have a different what you call is chemical properties. So these are the glucoses, fructoses, as well as galactoses. Okay, now I'll be showing you. So I think we have seen about the condensation process by which it is being released, the water is released, and they have formed up the what you call is a sort of like the disaccharides were being formed here. We have seen glucose forming up the maltoses. We have seen, coming back to the classification here, let me show you that we have seen that the monosaccharides are having a single units. Okay, and these are having the same chemical formula, but they are going to differ in the arrangement of the structure. That is, arrangement of the atoms is going to differ, which is going to lead to the what we call is the different chemical properties which are being present. Okay, now coming on to the other category, which is going to be disaccharides. Okay, disaccharides, they have the di where it means the two, and the saccharides meaning the sweet, but that is, these are going to have 2 units of it. Like we have seen that these sucroses are going to have 2 units of these glucoses. Okay, let me show you these let me show you. You will get an idea of this. Yeah. So here you are able to see that sucroses are being formed by the combination of the monomers like glucose as well as fructose, the fruit sugar. Okay, as we told that these will be undergoing a condensation process. They will be losing a water molecule. They will be bonded by a glycosidic bond. Since this bond is in between two sugars, we call it as glycosidic bond. This is actually a covalent bond here. And coming on to the lactoses, which is yeah, called as milk sugar found in your milk, is going to be made of the monomers as that is galactoses as well as glucoses once again, and they are also being bound by a glycosidic bond. But here the need to show this image to you all is to just show you how the bond orientation has got changed. Okay, the bond orientation has got changed in the sucrose as well as lactose. Usually we see that sucroses are being hydrolyzed in your digestive system. They are being digested into the monomers back again. They are going to, because yesterday you have seen that hydrolysis is a process where addition of the water and the polymer or dimer is going to get separated into respective monomers. So here by the addition of the water, what we can see is the glucose separates from the fructose over here, that is sucrose. So the one which does this hydrolysis is going to be an enzyme present, which is being called as sucrases. So I will be specifying sucroses are hydrolyzed by sucrases, where ACE, where the suffix is ASE. ACE is going to be an enzyme, and all the enzymes are proteins. Okay, sucrose, all O's are the carbohydrates. Usually your carbohydrates are going to end with a suffix of O's, and enzymes are going to end with a A's. So your sucrose is going to be formed by the condensation of glucose fructose, and sucrose is going to be hydrolyzed into respective monomers by utilization of an enzyme called as sucrases. Now coming on to lactoses, I have to speak about this sugar present in the milk, and you can see that the orientation of the glycosidic bond in both of them varies a lot, and the enzyme utilized to break down this is going to be lactases, and the sugar is going to be lactose. But as a person ages, they lose the secretion of lactases. Okay, the amount of the lactases are going to decrease, and since there is lack of lactase enzyme, the lactose cannot be digested or cannot be hydrolyzed into respective monomers like glucose as well as galactose. I think you're getting it. The absence of the lactase enzyme is going to fail up with the hydrolysis of the lactoses, which is going to lead to the accumulation of lactose inside the gut, which leads to stomach aches, and such a condition or pathological condition is being called as a lactose intolerance. Okay, I think you have got it. The reason to show you these dimers is, that is these disaccharides, is to speak about the glycosidic bond linkage, how the orientation has got changed in both of them, and they require their own respective sort of enzymes. Okay, we have to remember about the lactose intolerance where there is absence of lactases. Okay, this is the one I was talking about. Let me come back to the classification. So glucoses are utilized in the cellular respiration. The fructoses, the galactoses, one of the main composition of your lactose sugar. Fructoses, these are the components of your fruits. Okay, these are the monosaccharides. There are no repetitions. They are the monomers by themselves, so we call them as monosaccharides. And the disaccharides are formed by the combinations of the monosaccharides. In disaccharides, there are two monomers linked up with a glycosidic bond, which is a covalent bond. We have seen about the maltoses, we have seen about the lactose, and we have seen about the sucroses. Let me see if I have got any maltose sort of what we call is, let me see. I think I haven't got any lacto, that is maltose structure or linkages. Let me check if I have any one of it. Let me come to it. No. So the one I have shown you is the same. There is the sucrose as well as the lactose. So children, please remember that the linkage and the orientation of the glycosidic bond plays a major role in giving up the specific chemical property to that particular saccharides. Okay, saccharides. Now coming on to the next, I think we have to check regarding the monosaccharides are these, but we can even classify our monosaccharides based on the presence of the number of carbon atoms. Okay, let me come to that such that you will understand the classification of the monosaccharides now. So monosaccharides can be classified based on the number of carbon atoms they are going to have, and if the carbons present here are going to be 3, it's going to be called as trioses, because I told you all the carbohydrates are going to end up with the suffix "ose." So triose. And one of the triose is glyceraldehyde. Glyceraldehyde. Do you remember where we have seen the glyceraldehyde? I'll give you a hint. Glyceraldehyde 3-phosphate. Where was that? It was in your cellular respiration, right? Glyceraldehyde 3-phosphate. And it is a 3-carbon atom compound formed from fructose or fructose 1,6 biphosphate, if you remember. So 3 carbon atoms. If a monosaccharide is going to contain, if a monosaccharide is going to comprise of 3 carbon atoms, then we are going to call it as trioses. And such an example is going to be the glyceraldehyde. For glyceraldehyde, and we see such a compound of glyceraldehyde 3-phosphate in our cellular respiration. So that is a one here present. That is a triose, a 3-carbon atom compound. Now coming on to the pentoses having the 5 carbon atoms, and we have the hexoses. These are 3 of them, the triose, pentose, hexoses. These are the most major monosaccharides. We will talk about these 3 of them. We have even the other sort of monosaccharides, which will be comprising 4 carbon atoms, 7 carbon atoms, but we are specifying on the trioses, pentoses, and hexoses. Okay, triose we have seen it has formed during the cellular respiration process. And coming on to the pentoses, let me speak about the ribose sugar, as well as we have another sort of pentose, that is going to be your deoxyribose sugar. Okay, I'll be showing you these. Yeah, so this is a deoxyribose sugar, and this is going to be your ribose sugar. Orientation is same, but the only thing that is going to vary here is the second carbon atom in the deoxyribose sugar is lacking a oxygen, whereas the carbon atom in the ribose sugar, that is a second carbon atom here, is having a hydroxyl group, so it's rich in oxygen, while your deoxyribose sugar in the hydroxyl group, there is no oxygen, so it's no more a hydroxyl group, so it has only a hydrogen attached to the second carbon. So since it is lacking oxygen, we call it as D, that is removal, oxyribose, deoxy, because the removal of one oxygen from a ribose sugar has formed deoxyribose, and this is your ribose sugar. So this is a pentose sugar because it is comprising of 1, 2, 3, 4, and 5 carbon atoms. So each, the ribose sugar making up the RNA, most important sugar composition of your what you call is a RNA, is a ribose having 5 carbon atoms, and the deoxyribose sugar of DNA having 5 carbon atoms. Both of them vary in one, that is the second carbon of your deoxyribose sugar lacks one of the carbons, so we are going to put it under the category of deoxyribose. I think you have understood that these are pentoses. Coming to the what you call is, they are actually the monosaccharides. They are the monosaccharides. I told you that monosaccharides are being classified, that is monosaccharides are being classified into the what you call is these trioses, tetroses, pentoses, hexoses. So triose, you have seen gap molecule, that is a glyceroldehyde 3-phosphate, which I usually call as a gap molecule. And coming on to the pentose, the ribose sugar and the deoxyribose sugars. Next we have is hexoses, that is going to be your glucoses. And when I speak about the glucose, I should even again talk about the glucose getting stored in form of glycogen in the liver, and glucose getting converted in the plants as starch, glucose getting converted as a storage molecule in the plants even as celluloses, but we do not have the enzyme to digest the celluloses. Okay, so you have seen that the classification we have seen of the carbohydrates, they are monosaccharides. Okay, they are the monomers, they are the dimers, and these are the actual polymers comprising of a long chain of polymers, that is long chain of monomers coming together by a process of condensation, releasing water, forming the strong covalent glycosidic bonds. And these are the polysaccharides, that is a starch, where we could tell you that the glucose actually is going to be the monomer, and repetitively bonding up with the covalent bonds, the glycosidic bond, forming starch molecules in the plants, where they are stored in the roots, where they are stored in the seeds, etc. And the fibers, what I spoke about, celluloses, where it is again formed by the repetitive units of the glucoses, that is a celluloses. And that this cellulose is present in your diet, we are going to call it as a dietary fiber. And this dietary fiber is going to actually add up the weight to your food material. Okay, it is going to help in a proper bowel movements and peristaltic movements. Coming on to the celluloses, can't be digested in the human gut because of the lack of the enzyme called as cellulases. Okay, and coming on to glucose, stored in the animal body as a storage molecule called as glycogen in the liver. So these are the storage molecules, storage energy storage molecules found in the plants, the starch and the celluloses. And the energy storage molecule present here in the animal is going to be the glycogen. So this is a classification of the carbohydrates. Okay, and let me show you the process, that is sucroses and lactoses. I think I have spoke about the orientation and linkages that I have shown you. These play a major role in giving them a respective sort of like chemical properties. Please do remember them. And next, coming on to the glycogen and celluloses, let me show. These are the two of the energy storage molecules found. If you can see here, we see that this is the starch chain, which is formed by the repetition of glucose as monomer, but see the difference between the starch and the cellulose. You should get a doubt that starch as well as the cellulose, both are the monomers, that is the polymers formed by the combination of the glucose monomers, right, by having this covalent bonds. Then what actually makes it different? Because I told you that if the structure changes, the chemical properties changes, right? So if you can see the orientation here, we see that in the cellulose molecule, every adjacent glucose molecule has flipped. If you can see, this oxygen which was towards the top was being flipped to the bottom, to the top, to the bottom. So they have got flipped, and they are compactly arranged. So they are going to be very strong, very, very strong celluloses. Okay, they are fibrous, they're very strong. This is just because of the compact structure as well as the flip structure, as I told you, that the orientation of these the components have changed over here. As I told you that this is on the top, this is on the bottom, here came back to top and the bottom. So the flip of the structure has resulted in the different that is polymers formed. Here it has formed a starch, and here it has come back to the celluloses. You can see glycogen, how it's varying. The glycogen here is going to be actually a branched structure. Let me, one second please. Yeah, you can see glycogen is forming a branch structure. So even the branch orientation changes its chemical properties and all. So you can see that both starch as well as the celluloses are the linear structures, whereas a glycogen is a branch structure, and the orientation has changed, so the chemical properties changes. Though all of them are going to be the polysaccharides, they are not equal to they are not equal, though they are going to have the repetitive or the monomers are the same, but the orientations have changed, the compact nature has changed. So few of them are linear, few of them are branched. This actually makes carbohydrates the most toughest molecules to actually crack down or to learn about the structures and the chemistry of these. This makes it very harder when compared to that of the proteins or the lipids, actually, because these are actually highly branched structures, the carbohydrates. I think I have given you this. Now coming on to the glycogen, let me come check it out. Yeah, the same image I have. So coming on to the glycolipids, that is, I spoke about the cellular structures, the plasma membrane and extracellular matrix, which is going to actually going to have these carbohydrates. You remember while I started the video, I spoke about. So now you can see this, that this is bilipid layer and these are the lipid tails. So with this lipid tail are the carbohydrates chained up over here, forming structures like glycolipids in the plasma membrane, glycolipids. And this is the white matrix over here whole together is being called as a extracellular matrix, and this is going to be intra, that is your cytoplasm. Okay, in the extracellular matrix you can see the glycolipid, that is a carbohydrate chain out of these lipid layers. And you can even see the carbohydrate layer here, which is going to be your glycoproteins. These are the proteins, these are the membrane proteins that we are going to have in the plasma membrane, and these are topped up with the what you call is carbohydrate chains here. The carbohydrate chains, these are the glycoproteins that we are going to have, which are actually acting as a what you call is a surface antigens, which are going to actually responsible for the identification of the cell over here. So these are the glycoproteins and these are the glycolipids that I spoke about. So you can I think you have to get a clear cut idea now that how carbohydrates are very important in cell signaling, and how important are they in making up the antigens, because every cell has got its own like specific antigen. Like your A blood group person has his own sort of like antigens which are not matching up with the B blood group or O blood group. So antigens are these glycolipids that I have spoke about. Okay, so now coming on to the important last topic that I was specifying that I'll be talking about this at the end of the video. So now coming to this is the important thing is here, the ones that you're seeing is the RBCs, and this RBC belongs to the blood group O, and this belongs to the blood group A, this belongs to blood group AB, and this belongs to the blood group B. Okay, you can see that every RBC is topped with the sugars here, that is carbohydrates. Now I have shown you that they are going to be on the plasma membrane with the bilipid layer, right? Those lipid tails, they are being embedded with them. So this is a glycolipid, a glycolipid. These are these surface carbohydrates as a glycolipids, that is antigens. I'll be referring to them as antigens. So here also you have on the B, there are some specific antigens on A and AB. Now let us see if you can see this big structures over here. They have actually magnified these structures. You can see the carbohydrate chains here. What is a common thing that you can see here is the O ones are having these four structures. Okay, even the B has that, right? Even AB is having that common sugars, right? Even A is having that common sugars here, the carbos. The carbos are the same. What is difference is, in the O blood group, there is lacking of a fifth carbohydrate, that is this, right? You can see here that AB blood group also, you can see that here this is a 1, this is a 1. Can you see that in the AB, B, there are common ring structures that are present in the blood group O. So the ones which are extra, the purple over here, okay? The A and the AB blood groups. Check it out. In the A and the AB blood groups, there is a specific carbohydrate present here, the sugar, and this is another sugar that is present here, which is being called as N-acetyl galactosamine. N-acetyl galactosamine. So there are possibilities that we can actually cleave off this bond, then you can actually transform blood group A into O and blood group AB into O. Actually, this is not a recent research, but it's been years that people are working on this. But uh, there are sort of enzymes which have been actually discovered, and the enzymes which are actually used to cleave this glycosidic bond are glycosidases. Lactoses digested by lactases, celluloses digested by cellulases, and that cellulase enzyme is only present in ruminants, not in humans. So we are not able to utilize the energy present in between. You remember when I spoke about the breaking of the bond, you need energy, it is endothermic. And when it is, so your breaking a bond is going to be an endothermic process. Now coming on to, I'm talking about the common sugars present over here, common carbohydrates present over here. So why can't we cleave off these extra ones to make these blood groups to be same as to that of this O blood groups? That can be possible by utilizing an enzyme which can break the glycosidic bond, and that enzyme is being called as glycosidases. It is being called as glycosidases. Let me actually put this here. So they will be called as glycosidases.